Welcome to this particular lecture on microbial hormone. In this particular lecture, I shall be focusing on the different hormones produced by algae. Algae, as you know, is a group of photosynthetic organisms which can be both microscopic as well as big macrophytes. Algae can be mainly classified on the basis of their pigments. The major pigment being chlorophyll, that is chlorophyll A, B, C, D, E, and there may be also other pigments in the form of phycobilin, in the form of carotene or xanthophyll or fucoxanthin, etc., which actually accounts for the different coloration of the algae. As because algae is both macro and microphytes, they are dividing and growing with the help of different hormones. And these hormones can be major or minor. The major hormones mainly help in their growth and development, while minor hormones help in their cell division or sometimes help in their colonization. The major hormones produced by algae include Oxen, Gibberellin, Cytokine, while the minor hormones may include the Abscisic Acid, the brasinosteroid, or they have different other polyamines, etc. To start with, we will see the action of Gibberellin. Now, Gibberellin is produced by different green algae, and this algae may be dendrobial or may be unicellular in nature, and Gibberellin is mainly produced from the primary compound, that is geranyl diphosphate, which is converted to kaurine and kaurinic acid, and finally it will produce the gibberellic acid 12, which can actually produce different sets of gibberellin 15, 24, 9, 7, or at the same time it can produce GA53, GA44, GA20, GA8, etc. So the primary compound in the production of gibberellin in algae seems to be kaurine, and the different enantiomers of kaurine will include or involve the enzyme of kaurine oxidase and also the kaurinic acid oxidase, which will ultimately produce GA12, and once the GA12 is produced, the different other gibberellins are produced in green algae. Now, sometimes these green algae in the form of minute cells may actually grow in the rhizosphere of rice plant and may also be growing in the grains. As you can see in this particular rice grain, there is a film of green algae growing. Now this green algae will produce the gibberellin, and this gibberellin will enter into the rice seed, as a result of which this gibberellin will stimulate the growth of the embryo. And so the rice seed will germinate faster, and so the dormancy period will be reduced. So this is because of the algal colony growing on the surface of the rice grain. Now, apart from this particular action, gibberellin may also help in other functions, like it will stimulate the growth of certain branches. The most important growth hormone in algae seems to be Oxen, and Oxen follows a unique pathway. As you know, the starting amino acid is tryptophan, and from this tryptophan it will produce, or it will undergo decarboxylation, producing the tryptamine, and the tryptamine will be converted to indole 3-acetaldehyde, which will be finally converted to indole acetic acid. And this indole acetic acid is the prime compound of Oxen present in algae, though there may be other possible ways of production of indole acetic acid from tryptophan followed in other groups of organisms. Primarily, this Oxen will help in the growth of macrophytic algae, as it is shown here in Cladophora. The growth of the axial branches or the axillary branches is primarily because of Oxen production. At the same time, Chara, which is a major macrophytic algae, the axial leaves are produced, and these axial leaves will be bearing the sex organs in the form of globule, which is the male sex organ producing the antherozoids, which will move upward and will be trapped by the crown of the nucleus, which is the female sex organ, and fertilization will take place. So the growth of the reproductive structure and will mark the reduction in the axial branches, and this is helped by Oxen. At the same time, there may be the brown algae fucus. In fucus, the growth, the dichotomous leaf branching is brought about by the indole acetic acid or Oxen. Coming to the next important hormone, which is kinetin or cytokinin. Now this cytokinin is also produced by a group of algae, and this algae normally may be the green algae, the brown algae, etc. But primarily this cytokinin is produced by the combination of adenosine triphosphate or adenosine diphosphate with DMAPP or dimethyl adenosine pyrophosphate, which will produce a series of compound, and primarily it will produce the zeatin triphosphate. And once the zeatin triphosphate is produced, it will produce the zeatin diphosphate, then zeatin monophosphate, then zeatin riboside, and then it will produce the zeatin, which is transzeatin. So as you can see, the transzeatin is the most common form of cytokinin present in algae. And in which type of algae? They may be present in the unicellular green algae Euglena. And actually, when the cytokinin concentration in Euglena increases, it will actually increase the rate of cell division, and the number of Euglena cells will increase. As you can see, the dividing Euglena cells. It is also seen in Synodesmus. The cells will be dividing horizontally, and the horizontal division will result in the production of a colony, and primarily the peripheral cells will develop the cilia or the flagella, which will help in the locomotion. So this is also because of the concentration of cytokinin in Synodesmus colonial algae. It is also present in macrophytic brown algae like in Macrocystis, where the primary concentration of zeatin will help in the elongation of the leaf. And so it is the balance of Oxen and zeatin which will help the elongation of the leaf, and as the leaf gets elongated, there will be more photosynthetic process, as a result of which more food production. So nutritionally they become stronger. Coming to the very interesting algae, which is the Ectocarpus. This common brown algae is commonly seen in marine environment, and this Ectocarpus can be having two types of plant body. One is the sporophyte and the other one is the gametophyte. Incidentally, the sporophyte and the gametophyte, they look the same. So naturally, this type of alternation is called isomorphic alternation of generation. And in both the cases, that is in the gametophyte and in the sporophyte, there will be a reproductive structure called plurilocular gametangium or plurilocular sporangium, which looks the same. But in addition, in sporophyte, there will be the unilocular sporangium, which will actually produce the gametophyte in future. So this type of alternation is followed. But who determines this alternation? It is actually a group of aromatic kinetin compounds, which is commonly known as topolin. So this topolin is also a derivative of zeatin, which may be orthotopolin or metatopolin, and they primarily will impart a unique smell in this group of algae, and thereby they can be useful in the form of identification of this particular algae. Coming to the next group of hormone, which will include the ABA or abscisic acid. Now, abscisic acid commonly is a minor hormone present in algae as well as in higher plants. ABA production is following a simple process of G3P pyruvate conversion. What is happening here? The G3P and pyruvate will combine to produce IPP, and that is farnesyl pyrophosphate, which will ultimately produce the lycopene and beta-carotene. And this beta-carotene will be converted to epoxycarotenoid compound in the plastids or in the chloroplast of algae, and will be converted to intermediates like violoxanthine and neoxanthine. And this violoxanthine and neoxanthine will condense together to produce ABA or abscisic acid. And this abscisic acid is unique in some filamentous green algae like in Spirogyra. As you know, the filament of Spirogyra will grow with the help of Oxen, but once the vegetative growth is completed, they will undergo reproduction, and this reproduction is by the help of the production of gametangia. What is gametangia? Gametangia is the gamete produced by the fusion of the entire protoplast. as a result of which one filament is the donor filament, other one is the recipient filament, and the gametangial fusion will result in the production of zygospore. So it has been seen that once the reproductive ability starts in this Spirogyra, the concentration of auxin falls and that of ABA increases. So in one way we can say abscisic acid actually limits the vegetative growth and will force the algae towards reproductive growth. Coming to the minor group of hormone which include the stilbene. Now the stilbene, as it is shown here, there are two hydroxyl group attached and there is also a unique double bond present in the ethene form, and that is actually called dihydrostilbene, and this dihydrostilbene is commonly called diaryl ethene. This diaryl ethene is produced from simple amino acids like phenylalanine. So phenylalanine and tyrosine are the starting compounds of this particular stilbenoids or stilbene compounds, and this will condense together to produce the para-coumaric acid and then the coumaryl coenzyme A, and finally they will produce the tetraketide which will be converted to resveratrol and chalcone. And this resveratrol and chalcone are commonly, they are known as defense related compound. What does it mean? It means if these resveratrol group of stilbenes are produced in greater amount, the algae will be protected from bacterial or fungal infection. And at the same time, this particular stilbene, if the concentration of stilbene increases, then they will be showing greater longevity or greater vigor of growth. As you can see, this is an enteromorpha where there is signs of withering, but when the stilbene content is higher, enteromorpha is shining, and that is the name, that is the Greek word stilbos meaning shining. Coming to another very important group of hormone which is commonly called linoleic acid. Linoleic acid is an organic acid which is derived from a group of bryophyte which is commonly the Linoleiria, as it is shown here. This Linoleiria cruciata is actually a terrestrial bryophyte which produces linoleirin or linoleic acid. But at the same time, this linoleic acid is present in different other unicellular or macrophytic algae like the Dunaliella salina, which is a unicellular green algae. It is also present in the golden brown cells of Haematococcus, which is a brown algae. It is also present in the brown algae laminaria. And this laminaria, as you know, is a common macrophytic marine algae. Now, what commonly is there? This linoleic acid is actually produced from dihydro-4-coumaryl coenzyme A, which is producing the pre-linoleic acid, and finally it will be converted to linoleic acid and linoleirin. So once the linoleirin is produced, what it does basically is this particular linoleirin will mark the evolution of bryophytes from algae. That means the terrestrial plants have been derived from algal source, and at the same time this particular linoleic acid will also act as abscisic acid, meaning the reduction in metabolic activity, growth and development and initiation of reproduction. Coming to the next important group of minor hormone in algae, which is called the Jazzmonic acid. Now this Jazzmonic acid is also produced by both unicellular macrophytic as well as macrophytic algae. It is present in Dunaliella salina, it is present in Euglena gracilis. Of course, when I call about Euglena, Euglena is sometimes also considered to be animal because of some animal features, but actually it is having chloroplastid, so broadly they are placed under green algae. It is also present in a red macrophytic red algae, that is the Gelidium latifolium, and this Gelidium is also having the storage food in the form of agar-agar, which is a neutral polysaccharide. Now this particular Jazzmonic acid is actually produced from simple amino acid which is the alpha linolenic acid, and this alpha linolenic acid will be converted to different other forms, the epoxy derivatives and the other derivatives in the form of AOC, OPR, OPC, and finally it will produce the jasmonate or Jazzmonic acid, and sometimes it will also produce the jasmonyl isoleucine. And what does it do basically? This particular jasmonate or Jazzmonic acid will trap a particular external stimuli and will help the organism to grow. And at the same time, this particular linoleic acid, which is an essential fatty acid, is also being produced within that particular algae as a result of this stimulation. And once this particular stimulation is over, the algae will show modification in gene expression. And because of the modification in gene expression, they can be tolerating insect or pathogenic invasion. And thereby we can say that the Jazzmonic acid, which is trapped by the SCF receptor, once it is received, the stimulation of jasmonate is started inside. So one way or the other, the jasmonate production increases, and once it increases, it will stop the enzyme which is required for jasmonate production, because adequate amount of jasmonate is already there. And this jasmonate will activate specific receptor in the form of TFS, and thereby the gene expression will result in tolerance to insect or other pathogenic damage. Coming to another unique group of minor hormone in algae, which are commonly called polyamine. Now, why it is called polyamine? Because large number of amino acids are binding together, having large number of amino groups. So these particular polyamines, they are also produced from both microscopic as well as macrophytic algae. They include the cyanidium, they include the great lupia, they include the dictyota, that is the brown algae. They also include the green algae, macrophytic green algae called Ulva rigida. Now, the biosynthesis of polyamine is again very unique. It can start with a common amino acid like methionine or ornithine. Ornithine is converted to a very common putrescine. Putrescine is converted to spermidine, and spermidine will produce spermine. But this particular production of spermidine from putrescine will involve the involvement of adenosyl methionine. So methionine in one way is condensing with the byproducts of ornithine, and finally the spermine is produced, and once the reaction is over, the methyl adenosine is a derivative or is a compound which is produced as a result of this pathway. These polyamines actually have a very unique role. These polyamines help in vegetative growth. These polyamines also can help in the tracing the evolution of red algae from green algae or vice versa. And of course, polyamine sometimes it helps in the development of thallus of macrophytic algae. And there may be, sometimes it is so called that this polyamine may act as a particular cation which will bind with the DNA, will modify the DNA function, will modify the translation, will produce certain protein which will help them to survive during unfavorable condition. And at the same time, it is said that it is marking the end of the resting period. That is, once the resting period is over, greater amount of polyamine will be produced and the algae will get back to its normal growth phenomenon. So that is a unique phenomenon which will mark the end of the normal resting period and will mark the beginning of the growth hormone. So in one way we can say that the polyamine is marking growth hormone production. Coming to the next important group of hormone, which is commonly called the brassinosteroid. Now this brassinosteroid, the name comes from brassica, which is a higher plant, and steroid is a steroid group of molecule. It is produced by a unique network-like algae, which is commonly called Hydrodactyon reticulatum. Reticulatum meaning a network. It is so called because the plant body looks like a network, unique network-like mesh. This particular brassinosteroid is also produced from simple campesterol or other group of steroid molecule, and they are producing innumerable number of steroid compounds within the algae. And this particular steroid molecules has got a unique function. Actually, they help in growth, they help in development. How? By actually collaborating with other growth hormone like the gibberellin. So gibberellin along with brassinosteroid will help in the growth and development both in higher plant as well as in algae, as seen in this particular figure. Coming to next group of hormone, which is commonly called Rhoda Morphine. Now this Rhoda Morphine is a minor hormone which is a glycopeptide having a very low molecular weight, which is only 14 kilodalton. And this particular Rhoda Morphine is produced by a red algae in a branched filament of Griffithsia. And what do they do basically? They help in the growth of this filament. So the increase in the rhodomorphine concentration will allow the increased protein synthesis, and which is brought about by rhodomorphine. This is one of the aspects. But another very interesting aspect is there. That is, if sometimes the algae, the filamentous algae will be suffering from mechanical injury. Due to this mechanical injury, the filament may get damaged, or the filamental cells, the intercalary cells may be destroyed. What happens is, the moment this particular intercalary cells is lost, there will be increased amount of rhodomorphine accumulation. And because of that, the cells will regrow, regenerate, and the filament comes back. So what we can see here, rhodomorphine production help in healing of wound and also regeneration in some groups of red algae. And coming to the last group of hormone, which is commonly a group of polypeptide. It has been isolated from a unique brown algae called the Fucus diasticus. This particular hormone is actually a polypeptide which is having a typical feature which is commonly known as the polypeptide of around 244 amino acid having a unique LRR, that is leucine repeat sequence. The moment I say this leucine repeat sequence will bind with a specific receptor, as it is shown here. The leucine receptor will be binding with this particular hormone, and they will be acting as a signal. And once this signal is transduced, there will be definite differentiation, growth, embryonic organization, etc. And this type of embryonic organization is also seen in case of Fucus. What we see is the unique cell, and from this unique cell, once the particular polypeptide signal is received, they will show polarization. Though this polarization is also brought about by calcium ion. And after this particular calcium ion is deposited, there will be the other particular actin microfilaments active, which will allow the growth of the rhizoidal cell. So from this Fucus, what we can see here, the polypeptide signal, the calcium ion, and the actin protein will bring about the growth of the rhizoid, which is ultimately acting as the root, and the upper part of the cell, as it is shown here, will produce the plant body. So thus we see that this particular polypeptide is a group of microhormone which is helping in the morphogenesis of a macrophytic algae. So in this particular lecture, what we have learnt is, though algae is a group of microscopic organisms, it sometimes can be macrophytes, and these macrophytes or microscopic organisms will produce different major hormones in the form of gibberellin, in the form of oxen, in the form of zeatin, and at the same time it can produce some negative hormones like abscisic acid. At the same time it can produce some unique hormones like the brassinosteroids, like the polyamines, like the polypeptides, and they will ultimately help in the growth and development of the algal cell. Thank you.